Good morning. Thank you very much for the invitation, for putting this program together, for Ann and I for hosting us. We heard about oligosaccharides quite a bit the first 2 days. The word popped up here and there, microbiome, prebiotic, oligosaccharides. So this morning we're going to really dive deep into what that means, human milk oligosaccharides, or short, HMOs. Okay, a couple of words about disclosures, where the funding is coming from, and then we go right in. And I start with a statement that we've seen a few times in previous presentations in the first 2 days. And this is from the editorial commentary in the Lancet Breastfeeding Series 2016 that said that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion. And for us, the question is, why is that? How does it work? What is behind that statement? What is in human milk that makes it so powerful? So if we lift the curtain behind this statement here and look at what is in human milk. What are the macromolecules that we find in human milk? There's proteins, fat, lactose as the predominant milk sugar, and then there's oligosaccharides. 5 to 15 gram in mature milk, which often exceeds the amount of total protein. And in comparison to bovine milk is 100 to 1,000-fold more than we find in bovine milk. And since bovine milk is the basis for most infant formula, there's hardly any human milk oligosaccharides in infant formula, at least until recently. We've got to come back to that in a minute. So the talk is about structure and function. And I'm going to keep the structure part as short and uncomplicated as possible. We have to talk about it a little bit. So it's not as dramatic as this picture shows it. When we think of sugar, we think of something like this. When we think of sugar in the infant space, we think of something like this. That's fairly complex to me already, looking just at the structure. But when we talk about human milk oligosaccharides, which are complex sugars, that's not the same. So it's not the typical sugar that you think of that you associate something bad with. It's actually something good. And I'm going to show you throughout the hour what potential benefits there might be of these human milk oligosaccharides. So here's a fairly simple structure of a human milk oligosaccharide. You don't want to draw this out every time you talk about this. So we give every monosaccharide building block here a separate symbol, and then assemble them together depending on the structure. And really, there's only 5 building blocks. It's glucose and galactose, which we know forms lactose. And then in addition to that, we have N-acetylglucosamine, fucose, and sialic acid. So 5 building blocks, and depending on how you put them together, you get different oligosaccharides. So it's a little bit like Lego bricks. And you can imagine if you put Lego bricks together, you either get a car or you get a little plane. And you can imagine these two things do different things, right? A plane does different things than a car. And that's the same for the oligosaccharides as well. That's fairly simple structures, but structure determines function. And it can get fairly complex. That's my daughter getting the 1,200 pieces Lego set here for Christmas. It doesn't get that complex, so it's not that bad. Okay, so let's take a look at oligosaccharides and different structures. All human milk oligosaccharides have lactose at the reducing end. It's glucose and galactose. And then they get extended by disaccharide units in different linkages. They can be branched, further elongated. So you see already this gets fairly complex, either very long or branched. You can modify these structures with fucose in different linkages. If you add fucose on lactose, you get 2-fucosylactose, 2-FL, and 3-fucosylactose. Or if you add fucose at the growing chain, you get different oligosaccharides. And here an example for these 4 oligosaccharides. They have the same monosaccharide composition, but you put those Lego bricks together in different ways, you get different structures. And we think that these 4 oligosaccharides indeed have different functions. Gets a bit more complicated from there. You can also add sialic acid. Sialic acid introduces a negative charge through its carboxyl group. If you add sialic acid to lactose, you get 3 and 6-sial-lactose. And if you add it to the growing chain, you get different structural isomers. You can add more than just one sialic acid, like down here in this structure. And the important part really is that there's about 150, 200 different oligosaccharides identified so far, and every mom makes different ones. So it's almost like a thumbprint. huge variation from mom to mom. I'm going to show you that in the next few slides. So what are we going to focus on? What's my lab focusing on? One is maternal drivers. What contributes to that variation in oligosaccharide composition between different moms? What's the benefits for infant health and development? And then can we learn from human milk and take some of that knowledge and apply that to develop new therapeutics outside the infant space for adults? So let's take a look at the maternal drivers first. If you look at this slide, this is data from the child cohort in Canada. It's about 1,200 subjects where we have oligosaccharide data for. And you see down on the bottom here, every bar is a different subject. And on the y-axis, you have in different colors the concentrations of different oligosaccharides. That looks very different. Right? There is low amounts in these samples, very high amounts in these samples. And even if you look at relative abundance, so you set it all to 100%, the pattern looks very different from mom to mom. So like I said, this is like a thumbprint almost. You can take a look at oligosaccharide data slightly different. This is a principal component analysis in the three-dimensional space. And you see here every dot is a milk sample. There's about 10,000 milk samples in this plot. And the closer those dots are together, the more similar the oligosaccharide composition. The less or the further apart those dots, the less similar the oligosaccharide composition. And just by looking at this, so this is collected from 10,000 samples, as said, approximately, and really from almost all over the world. So most of the samples come, of course, from North America, where we are located, but we have almost every continent covered and many countries included. So you can see already this distribution here in these two clusters, right and left. What is that? Is that two different types of moms? Why is there such a separation in the oligosaccharide composition? If you go through this and separate it by where the samples were collected and which cohort they come from, you see, for example, here the Canadian child cohort in red, and then in comparison, a rotavirus project collected in India in green, and there is some separation, like in top down and top and bottom, but that doesn't explain the left and right separation. If you separate them by a different characteristic, you can actually see, that explains that separation. What is that? That is what we call the secretors and the non-secretors. What does that mean? So that is genetically determined specific mutation in enzymes or in genes that encode enzymes that add fucose to your oligosaccharides. So it's shown here in the corner. There is an enzyme here called fucosyltransferase 2, or FUT2, that adds fucose specifically in this alpha-1,2 linkage. There's another enzyme, FUT3, fucosyltransferase 3, that adds fucose in this alpha-1,4, alpha-1,3 linkage, depending on the underlying structure. So it happens that there is people among us that have a specific SNP, or specific SNPs, so that's single nucleotide polymorphism, that inactivate this gene. So that enzyme is completely dead. It's a mutation that introduces a stop codon. And these moms, or humans in general, can no longer add fucose in this alpha-1,2 linkage here. They still have a FUT3, and that explains the difference between these two different clusters here. It's one base pair. One base pair in the genome that changes the entire composition of human milk oligosaccharides. It's not just one oligosaccharide that's now missing or in a low or high abundance. The entire oligosaccharide profile is completely different. One single nucleotide polymorphism. So the secretors here have an active FUT2, and the non-secretors have an inactive FUT2. And you can map this out by different cohorts. This is, again, data from the child cohort. In red, you see the homozygous SNPs. Orange is the heterozygous SNPs, and white is the wild-type, two wild-type alleles. You see how that clusters out. So that's your secretor and non-secretor. You can do the same thing with FUT3. So that's the fucosyltransferase 3. And you can see there's clustering as well. This time you have the dark spots, the homozygous SNPs, down here in the bottom, orange somewhere in the middle, and white on the top. So you can draw a line like this. So eventually you can cluster this in secretor positive, Lewis positive in this quadrant here. So that's active FUT2, inactive FUT2 and active FUT3, and you can go through the different quadrants, and you see how genetics really determine the oligosaccharide composition on a global scale. This is not just one oligosaccharide. So we knew about FUT2 and FUT3 already, but we can take this a step further now and look through different oligosaccharides and how they're associated with different enzymes, and then try to understand which enzymes really are participating in the synthesis of human milk oligosaccharides, which we don't know yet. There's still an entire textbook page in biochemistry that needs to be written. So here, for example, we have a sialotransferase ST6-gal1 that you don't really see contributes to any of the clustering here, but you see there is individual oligosaccharides that are strongly associated with SNPs in this specific gene. And here specifically you see 6-sialactose, which this enzyme here catalyzes the reaction of adding sialic acid in a 2,6 linkage here. And you see it's not a loss of 6-sialactose, it's actually an increase of 6-sialactose when you have that SNP. So there's a gain of function SNP. And that does not relate to 3SL. It relates to a similar oligosaccharide, that's the extended structure, LSTC, but not LSTB. So there's very clear associations between different SNPs and different genes and oligosaccharide synthesis. Genetics play a major role in oligosaccharide composition. So now we're using GWAS, so genome-wide association studies, transcriptomics, HMOmics, and then use in silico pathway modeling and eventually in vitro target validation to unravel the pathway of human milk biosynthesis. Again, it's not known how most of these oligosaccharides are synthesized in the human mammary gland. And that changes depending on where you are on the planet. For example, if you look down here in Peru, or really in South America in general, most moms are secretors and have an active FUT2. 98%, 95% in other countries. That's different on the African continent. You find 20, 25, 35, up to 40% of the moms that are non-secretors. They don't make these specific oligosaccharides. They make up other oligosaccharides, but their oligosaccharide profile is completely different. And that really makes you wonder, what do oligosaccharides do to the infant, to the mom, in different environments, in different parts of the world? Oligosaccharide composition changes over the course of lactation. You see, for example, 3-sialactose goes up in concentration, 6-sialactose goes down, DSLNT goes up, and you can go through multiple different oligosaccharides, and you go through multiple different cohorts, and you find the same trends in different cohorts. It also seems to change depending on the season. So we found in the Canadian cohort, and others have found that in a cohort in Gambia before, that there is specific oligosaccharides that change concentration depending on where you are in the season. It can be very extreme, dry season and wet season in Africa, but it also happens in Canada. So we don't really know why that is and what contributes to that, but there seems to be seasonal variations as well. We've also seen that oligosaccharide total amount is different whether mom had already a baby or not. So the more infants were already here, the higher the oligosaccharide concentration. That is not depending on age, so maternal age is not associated with oligosaccharide composition. And mode of delivery isn't either. It doesn't matter whether you deliver vaginally or by C-section. For the oligosaccharides, that doesn't seem to make a difference. So that's the genetics, and I think there's a lot of questions that we need to answer in that part still. But there is many, many more other questions when it comes to maternal drivers. For example, what about diet and exercise? Can we manipulate oligosaccharide composition that way? We don't know. We have some data here from a mouse model. Mice make oligosaccharides, 2 to be precise, mostly 3SL, a little bit of 6SL. And mice are much easier to work with when it comes to changing diet and putting them on an exercise regime. So you can measure 3-sialactose concentrations in mouse milk and then put the mouse on a high-fat diet. And you see 3-sialactose concentration goes down. You can exercise the mouse, and the 3-sialactose concentration goes up. And you can do the cruel experiment to put a mouse on a high-fat diet and then exercise it. That's how I felt this morning, really. And you see that those two effects even each other out. Now you're back to almost normal to where you were before. Of course, this is mice. We don't know if this happens in humans. We have some data from a collaboration at Baylor, also from a collaboration in Arkansas, that points in the same direction. Silated oligosaccharides seem to respond to high-fat diet, but also to exercise. So we're working on that right now to confirm that, but it seems to hold up. Then we have some data that showed that using probiotics during pregnancy changes the oligosaccharide composition during lactation. What about maternal health? Gestational diabetes, chronic inflammatory diseases, how does that affect oligosaccharide composition, milk composition in general, really? We don't have a lot of data around that. How about using medication? How does that change oligosaccharide composition? No data whatsoever. So lots of question marks still when it comes to maternal drivers. Why is that so important? Why do we need to know what changes oligosaccharide composition? Well, if we identify that there is specific oligosaccharides or specific oligosaccharide profiles that are potentially beneficial for the infant or for mom, then we want to know how that actually works. How can we manipulate that? Can we put mom on a specific diet or on a specific supplement to enrich for specific oligosaccharides that we identify to be beneficial? So in the infant health and development space, before we get to that, we need to know what happens to the oligosaccharides when baby ingests them, right? So oligosaccharides go in, they're resistant to the low pH in the stomach and to the pancreatic and brush border enzymes. About 1% gets absorbed in the small intestine, and the rest either gets degraded by bacteria in the colon or appear intact, excreted in the feces. And you see this huge variation here, 10 to 80% excreted. That's saying it's either everything or it's nothing. It's completely meaningless if you think about it, but really it depends on the stage of lactation. Very early on, you find a lot of oligosaccharides that come out intact. Then the microbiome somehow establishes and learns how to deal with these oligosaccharides. And then you find a lot less oligosaccharides coming out intact because now your bacteria is starting to metabolize them. And then towards weaning, when you introduce other foods, then you find more intact oligosaccharides, but also other glycans popping out in the feces. So it depends a little bit on where you are. That's why there's such a wide range here. So what do these oligosaccharides do? I'll give you this one cartoon here and go through this quickly. We always talk about human milk oligosaccharides as being prebiotic, right? So that's the food for bugs. And that might be true, and there might be a lot to it. And we started what we call Mommy Biome, which is a combination of the Center for Microbiome Innovation led by Rob Knight in San Diego and our Mommy Core. So we really try to understand how human milk oligosaccharides and other milk components impact the microbiome. But we think there is so much more to it. We think HMOs are way more than just food for bugs. We know that they're antimicrobials, so they directly act on the bad guys and keep them in check. We know they're anti-adhesives. I'm going to show you a slide on that in a minute. They directly modulate epithelial cells. And they also impact the immune system, either locally in the gut or on a systemic level. Remember, 1% is absorbed. Sounds like it's not a lot, right? 1% is neglectable. But 15 grams going in per liter, and this is every 2 to 3 hours. The kinetics tell us that there is decent concentrations of oligosaccharides in the infant blood. We find it in maternal circulation as well. So there's local effects in the gut, and then there's systemic effects. And we believe we are here. We've seen this iceberg before. We think we're just really now understanding what these oligosaccharides do, where they go, what target organs there are, and what mechanisms are behind this. So there is a lot to discover in this space still. Let's talk a little bit about anti-adhesives, and that's going to be my first story on infant health. What does this actually mean, anti-adhesives? So we have this pathogen here that approaches the intestinal epithelial cell, and to be able to hold on in this very dynamic milieu, they need to somehow attach to epithelial cells. And you can imagine if you're a bacterium, you're swimming around in the intestine, lots of movements, you want to hold on to something because otherwise you're out pretty quickly. So the way they hold on is by interactions between bacterial lectins, so that's glycan-binding proteins, and glycans, sugar molecules on the surface of your epithelial cells, the glycocalyx. It's a thick layer of glycoprotein and glycolipid. And that is important for bacteria to be able to proliferate and in some cases to invade and cause disease. Now, if we go one step back and look at the same diagram here, but now in the presence of human milk oligosaccharides that really come in constantly in very high concentrations. And it turns out that these human milk oligosaccharides resemble some of the epitopes that we find on the glycocalyx. They serve as soluble decoy receptors, bind to the bacteria. Bacteria can no longer bind to your epithelial cells, and they get flushed out. So that's why we believe that these anti-adhesive effects of oligosaccharides protect us from some of the infectious diseases. There's one example here that we've done with Lars Ekman at UCSD, who sits right next to me in the office, funded by the Gates Foundation, where we specifically look at EPEC attachment. So that's enteropathogenic E. coli. And we found in tissue culture that in the presence of human milk oligosaccharides, EPEC attaches less to epithelial cells. Attachment is not a very good measurement for disease potential. EPEC specifically causes these lesions on epithelial cells that you highlight here in light green. We can measure those lesions. So that's the damage that EPEC is doing on your epithelial cells. And in the presence of human milk oligosaccharides, there's significantly less lesions on epithelial cells. This is done in tissue culture. We're now repeating this in organoids as well, human organoids. And also we've shown that this works in neonatal mice. So you can add oligosaccharides to the diet and EPEC colonization drops significantly. So that's one example. And you see here diarrhea is a major problem, 2,200 deaths almost per day, every single day, children under the age of 5. EPEC is not the most common pathogen for that. Rotavirus is one of the major causes for that. So we looked at rotavirus infection as well. We worked with Sashi Ramani here at Baylor in the Maria's test group, and specifically at a rotavirus called G10P11, which is the spike protein that sits on this specific rotavirus that is found in areas in India, specifically in the neonatal age, so the first 4 to 5 weeks of life. And we thought, well, anti-adhesive must work here too. Glycans are involved in the binding. Let's test it out and show it, get it published and out. So Sashi grew epithelial cells, added the virus, looked at infectivity, and to our surprise, we found that there is specific oligosaccharides that increase the infectivity of this specific rotavirus strain. And we thought, oh, this can't be right. This assay must be wrong because human milk is supposed to protect us. So how can this be right? We must be doing something wrong. So we repeated this a few times, different cells, different conditions, and we found the same results. We wanted to confirm this and went to India and looked specifically at the moms and the infants there, collected milk samples, analyzed them for oligosaccharides, and looked which infant developed or was infected with rotavirus and who had symptomatic and asymptomatic infections. And is there a difference in the oligosaccharides? And we found there is a difference in the oligosaccharides. This is looking at it more on a global scale here. This is principal component analysis. And you see already in red is the infants that are infected and show symptoms, so the milk oligosaccharide composition in corresponding moms, and they cluster slightly different. And then if we look at individual oligosaccharides, we find the same oligosaccharides that we found to increase infectivity in the in vitro assays were higher in the milk of moms where the infant got infected and had symptoms. So there was a clear overlap between the in vitro data and what we found in the mother-infant cohort here in India. So that was a surprise, to say the least. Is it possible that there are pathogens now that are getting ahead in the arms race and are trying to take advantage of human milk components? If that's the case, we need to know that. And can we use that knowledge then to get ahead, to develop better vaccination strategies? So it turns out Rotovac, which is the virus that is the vaccine that is used in that area, is a live attenuated strain, and we can increase the infectivity of Rotovac as well with the same oligosaccharides that showed an increased infectivity in the regular strain that we find. So can we add oligosaccharides to vaccination strategies and make vaccinations more efficient? So to summarize this part, specific HMOs are associated with increased rotavirus infection. And again, this is a specific rotavirus strain. This is not for all rotaviruses. And this is an opportunity to improve vaccination success. Another example is on growth. Are oligosaccharides related to infant growth? We had shown that with Michael Goran's group at USC, that there are specific oligosaccharides that somehow associate in cohorts with infant growth, fat mass, and all those parameters. And here we work with Kim Michelson's group in Copenhagen. He has this very unique cohort of exclusively breastfed infants with excessive weight gain. So if you look at the characteristics of these, there is a high weight gain group, and there is the weight for age z-score over 2 standard deviations. So these are gaining weight extremely quickly. And then the normal weight group here is between -1 and +1 standard deviations. And we wanted to know, is there a difference in the oligosaccharide composition between these two different groups? Can oligosaccharides explain this immense excessive weight gain in these infants? So we looked at total oligosaccharides. There was no significant difference in total oligosaccharides between the two different groups. We looked at HMO-bound fucose as a total measurements of fucosylation. There was a significant difference, more fucose in the high weight group. We also looked at HMO-bound silation, and there was no significant difference, although there was a trend in lower silation in the high weight group. And then we looked at specific individual oligosaccharides, and we found that specifically 2FL, 2-fucosyl-lactose, was significantly higher in the high weight group. Neo-LNT was significantly lower in the high weight group. So we have different oligosaccharides that seem to be at least associated in different ways, in this case with high weight gain. Now this is an exploratory study. It was only 30 mother-infant pairs, very small cohort, although we got significant results. And we wanted to confirm this. Well, let's go, let me put this here first. So this is now on a linear scale, 2FL and Neo-LNT. And just to give you an idea where we are currently with adding these oligosaccharides to infant formula, here's your 1 milligram per ml threshold. So we're way below that, way below the ranges that we measure in this study here. And Neo-LNT is right in the middle. And so in case you were wondering, are we now adding 2FL? Our kids are going to get fat now. We are way out of that range. Right? So at this point, no reason to be concerned. So that was 30 mother-infant pairs. We worked then with Hanna Lackström and Dr. Radava, who spoke yesterday. Now went to a Finnish cohort, the STEPS cohort. These are fairly healthy infants, now 5, 6 years old. And we had a chance to look at 800 milk samples from 800 mother infants. And we found the same thing. And this is presented a little bit differently here. This is the first 5 years of life. Here is your height standard deviations. And you see that the highest quartile in 2FL concentration gives you a higher height. And the lowest quartile gives you a lower height. So the same that we found earlier, the more 2FL you have, in this case the higher you are, but this is the same for weight as well. And the less 2FL you have, the smaller you are. It looks the other way around for Neo-LNT, just like we've seen in the Danish cohort. And if you take the ratio of 2FL and Neo-LNT, you see that this holds up nicely, and it holds up until 5 years of age. This is significant until 5 years of age. So you can imagine, depending on what oligosaccharides you give and what the ratio between different oligosaccharides are, you get the tall Scandinavian, or you get the smaller German guy, that would be me. And there's a difference of half a standard deviation, just by looking at the 2FL Neo-LNT ratio. I think this is quite remarkable. So again, specific HMOs are associated with infant growth, infant and childhood growth. We need to study causalities. Is this just an association, or is there really a causality behind this? How does this work? And this could be a great opportunity to improve growth when it's needed. Another example is in the premature space, just one slide, although this is work that now has covered 10, 12 years of the work that we're doing in the lab. But we try to summarize this in one slide here. Briefly, what is NAC? And why is it important? 5% of all very low birth weight preterm infants develop this often deadly intestinal disorder. We know that preterm infants who receive human milk are at 6 to 10-fold lower risk to develop NEC. Is there a connection with HMOs? Can HMOs explain this difference? So we set out about 10, 12 years ago to test this hypothesis that human milk oligosaccharides are somehow related to the lower incidence of NEC. What we did is we isolate oligosaccharides from pooled human donor milk to get the entire collection of human milk oligosaccharides, test that in an in vivo model, in this case in a neonatal rat model. We found that there is an effect, and we wanted to know which structure is it. So we went in, used multidimensional chromatography to really narrow down which structure is responsible for the effect that we see. And we found that this is the oligosaccharide that was responsible for the protective effect. It's called DSLNT, disillyl lacto-N tetrose. Then we presented this work, and we got slashed a few times saying, well, this animal model is not really good when it comes to necrotizing enterocolitis. You know, this has nothing to do with NEC, really. We looked at the chemical space around DSLNT, so we did a few studies, worked with the chemists to make derivatives, and really found that DSLNT was the most important component to be effective here. But we wanted to get around that criticism that this is just an animal model. So what we did is we designed a mother-infant pair cohort where we recruited specifically mothers and their very low birth weight infants in North America, collected milk samples every second day of the milk that went into that infant on that day, analyzed the human milk oligosaccharide composition, and compared oligosaccharide composition between infants that developed NEC and infants that did not develop NEC. And we found, to our surprise, that the same oligosaccharide that was protective in the animal model was lower in the milk that was given to those infants that later developed NEC. So it was protective in the animal model. If it was in low concentrations in mom's milk, that infant would develop NEC. So it was a clear match. We've repeated this in a study in South Africa in the meantime and found the same oligosaccharide being associated. Here we can say, well, it's an association, it's not causality, but since we have the animal data as well, there's a nice overlap that now gives us more confidence that we can move forward to a clinical study. And it also gives us some confidence that the model is not that bad after all, so studying the mechanism with the compound will give us some insights how this really works. So this is an example in the premature space. One specific compound. How are we doing with time?
Fine. 37 minutes. That's fine.
Okay, good. Give 10 minutes maybe to talk about something completely different. I know this seminar is about the neonate, impact on the neonate, but we have very exciting data to show that what we learn from human milk oligosaccharides in the infant space, in the neonatal space, can be very valuable when we go in and develop new therapeutics for adults at the other end of the life spectrum. So when we talk about frailty, atherosclerosis, arthritis, those kinds of things, we think that oligosaccharides can be very powerful in that space as well. I'm going to show you some data on that. So this is about chronic inflammation. It all started around 2014 when I went on a hike with a friend. This friend here is Philip Gortz, who's also a professor at UCSD, this guy over here. And the hike is up to Potato Chip, as we call it, in San Diego. And, this is the chip that you're sitting on here. My daughter takes that very literal here. And so Philip is working on macrophages, chronic macrophage inflammation. He said, Lars, we need to find a way to work together. I would love to test your oligosaccharides, throw it on my macrophages, and see what happens. So we did that. Again, isolate all oligosaccharides from human milk, and threw that on his macrophages, and we found, wow, it's a significant reduction in IL-6 and IL-1 beta when you stimulate the cells with LPS and then throw your oligosaccharides on it. We then used the same approach to identify specific oligosaccharides that are responsible for that effect, and we found that 3-SL, 3-siallactose, is the oligosaccharide that drives most of these effects. We found that this works in a mouse cell line. It works in primary cells. It works in human cell line as well. And it works in human patients as well. And we've shown that this is not just working on the RNA level, it works on the protein level as well. So the secretion of these cytokines is significantly reduced. We then went in and did dose finding, IC50 study. So we found the IC50s around 20 microgram per ml, which is close to what we have in the infant blood. It's not an unreasonable concentration. And we wanted to know, what is the mechanism? How does this work? How is 3SL reducing these very prominent pro-inflammatory cytokines? And to make this story short, I'm going through the next slides quite quickly. I'm going to show you one summary in the end. So we wanted to know, what's the receptor? Is it somehow interacting directly with the TLR4 receptor for LPS? And we found that's not the case. So we did RNA sequencing. We found that we stimulate the cells with LPS. You get a bunch of genes up and down regulated. But it's not the same genes that you affect with 3SL when you add that to the mix. So there's only a few genes that are differentially expressed in the presence of 3SL. So very unlikely that there's a direct interaction on the receptor, otherwise you would shut down the entire program. Then it's a sialylated glycan, so we thought, well, maybe it's the Siglecs. And we have the Siglec experts, right, one floor down, Ajit Barki and Victor Nizet. So they had knockout mice already running around. We just needed the bone, get the macrophages isolated, and see if our compound still has an effect. And it turns out it still works. So knocking out those Siglecs is not having an effect. Siglets are not the receptors. So what is the receptor? And now it really becomes a fishing expedition, right? Could be anything. It's very difficult to narrow down glycan-protein interactions. The affinity is not very high, so to do pull-down experiments, mass spec, very difficult to do that with a glycan. So we went back to our RNA sequencing data here, and we looked which genes are actually downregulated with 3SL. And not a surprise, it's the genes that are associated with inflammatory response, cytokines, cytokine activation. Which genes are upregulated, and we found, oh, that's interesting. There is a bunch of lipid metabolism involved here. And we found that there is all these genes that are associated with what we call an accelerated TLR4 resolution. So when you switch on a macrophage, you need to switch it off. That's the resolution phase. The faster you can switch a macrophage off, the less inflammation you have. So it turned out there was a paper published just a year before we looked into this from a guy at UCSD, Chris Glass' lab, that showed that TLR4 signaling switches on your inflammatory response here very early on. That's 1 to 6 hours. That's when all your cytokines are released. That's your active macrophage. But then it gets switched off by this pathway down here that includes a lot of lipid metabolism. And that happens in the 12 to 24-hour range. And it turns out that almost 90% of the genes that we saw induced by 3SL have something to do with this pattern here. So we went to Chris, asked him, can you help us with this? Can we throw our compound on it and then do ChIP-seq? So we did this experiment here, just to cut this very short now because it's very detailed, but just to show you that we're really going deep into this. So if you look at 2 hours, just LPS, you see all these NF-kappa B, all these different inflammatory pathways go up. But here you see the lipid metabolism coming up when you add 3SL to the mix. So let me skip this one here and go right to the model. Make this a little bit faster. So what we think is going to is happening, let me skip this one here as well. What we think what's happening, I'll skip this one here as well. That's what happens when you have too many slides. Okay, so here's the model. What happens when you add LPS or another stimulator? You switch on the inflammatory response and you get to this point here, right? Inflammation is switched on. This is your early phase of macrophage activation. While we still don't know what the receptor is, 3SL somehow interferes with this program here, shifts the macrophage from activation to resolution much faster. What happens in 24 hours now happens in 6 hours, and the macrophage gets switched off much faster. The lipid profile changes rapidly, much earlier these lipids pop up. We see the receptor for it that we can knock out. Now gives us that switch off. So the macrophage is switched off much faster. Now, why is that important? One of the diseases where chronic inflammation, macrophage inflammation, plays a significant role is rheumatoid arthritis. 1% of the world population has RA. That's about 1.3 million people in the US alone. And women are affected 3 times more than men. But they may also hold the secret on a potential therapeutic. So we collaborated here with Mary Patcourt and Nunzio Bottini at UCSD and tested out our 3SL in a RA model, in a mouse model. We put 3-sialactose in the drinking water. First, to show you, we measure the paw thickness here throughout the time of disease. We induce the disease by giving it a collagen antibody. Paws swell up. You give it an LPS boost, and the paws get really inflamed. Now you add 3SL in the drinking water, and you see there's a significant difference in the paw swelling. A lot less paw swelling. And if you show this to someone who is in the RA field, will tell you, wow, this is what we see with our TNF-alpha inhibitors. This is very comparable. And not only that, at the end of the study, you see there is less inflammation, less bone erosion, less cartilage damage. So all those readouts that are typical for arthritis are significantly reduced in the presence of 3SL in the drinking water. Right? This is not an injection. This is just giving something in the drinking water. So we tried this out in the second model. This is a genetic model. And just to make this short, we find the same in 3SL reducing the paw thickness significantly. In the meantime, there's a group in South Korea that used a third independent model, also tried 3SL, and also found that 3SL significantly reduces arthritis. And that's about the same concentration. So here now we have data from an in vitro model, a cell model. We have data from an in vivo model, actually 3 independent models where we show that this compound works. Is there a human cohort where we can see if this really translates to human disease? Now, that's difficult to do because most of our infants don't develop arthritis, of course. But when we showed this data to Christina Chambers at UCSD, she said, well, that's interesting because we have so many women with RA that go in remission at the end of the first trimester, and then they flare up again towards the end of breastfeeding. It's like, well, that's interesting because it turns out that human milk oligosaccharides, specifically 3SL, pops up in maternal circulation at the end of the first trimester during pregnancy. So is there anything, is there a link between these two things? So what do you do if you want to do a cohort study? You go to Canada, you go and call Megan Azad and ask, did you ask your moms if they had arthritis? And it turns out that they collected data on arthritis. They asked moms, have you ever had RA? And if they said, no, we had never had that, we call that 0. That's our control group. Have you ever had RA, but you currently don't have RA? So that's our remission group. That's our group 1. And then, do you currently have RA? Still have RA while you're actually giving us a milk sample? That's our active group. And then we looked at the oligosaccharide composition, and we found, indeed, no difference in total oligosaccharides, but significantly more 3SL in the moms that went in remission. So a very nice correlation now between the in vitro data in tissue culture, mouse models, and coming in with a cohort like this where you show that moms that go in remission make more 3SL and produce that in the milk. So 3-sialactose may be a potent therapeutic to treat and prevent arthritis. But there's other diseases that are related to chronic inflammation, chronic macrophage inflammation, and one is cardiovascular disease, the #1 killer in the world when it comes to annual death here. 18 million people die from cardiovascular disease. That's your heart attack and your stroke every single year. And macrophages play a huge role in that. That's your foam cells, right? That's the foam cells that suck up all your LDL, all your lipids, and then clog up your arteries. So to make this story short, again, working with Philip here, we put mice that had a LDLR knockout, so that's your receptor knockout, put it on a high-fat diet with and without 3SL, And then we looked at atherosclerosis assessment. No difference in food intake, no difference in weight gain, but a significant difference when it comes to atherosclerotic plaques in these animals on 3SL. And you can measure that in 3D. You can measure plaque size and necrotic course and all these things. And again, there's a significant difference in all those measures when the mice are on 3-sialactose. So can we take a human milk component and apply that to patients that would potentially die from a heart attack or a stroke? A natural component that we give to babies every 2-3 hours anyway, potentially safe. This could be a real game changer. So summary here, 3-sialactose may be a potent therapeutic to treat and prevent cardiovascular disease. That's just 2 diseases, really. RA, cardiovascular disease, I talked a little bit about diarrhea. There's so many other things, infectious superbugs, obesity, diabetes, IBD, breast cancer. There's always a link somehow to human milk and to breastfeeding. And we want to identify how this all works. And we call that our milk moonshot. We want to know what is in human milk, how this works, and how it could be protective for the infant, for mom, but also for adults that are at the very, very other end of the life spectrum. I keep you with that and take questions.
Dr. Mustaki first.
Carbohydrates in any situation can be, as you mentioned, inhibitors of adherence of microorganisms to receptors on epithelial cells. Surprisingly, unless I missed something, one of the important carbohydrates is mannose, high mannose type, oligosaccharides, and linked to asparagine residues. You don't have any mannose there.
No mannose.
So can you explain why there is no mannose, first of all? And second, sialic acid, alpha-2,3 or alpha-2,6? Because again, that makes a great difference with respect to recognition by influenza virus and so
Absolutely.
on, as inhibitor, that is.
Yeah. So no mannose in human milk oligosaccharides. Mannose is specific to your N-glycans. We don't find it in human milk oligosaccharides at all, which might give away a little bit how they're synthesized in the mammary gland.
But you find it on glycoproteins.
You find it on glycoproteins,
Oh,
you find it for sure in the milk, but you don't find it in human milk oligosaccharides. Yeah.
Okay.
And the other question was about the alpha-2,3, alpha-2,6 linked sialic acid. We find both.
Both.
Yep.
Okay.
And in different, interesting here is you have an alpha-2,6 linked sialic acid not only on the terminal galactoses but also internally on glutamac, and that is very unique to human milk oligosaccharides, really. Yeah.
Dr. Levchich first, and then Dr. Ogra. Carlos.
Congratulations, it really was a fantastic presentation and excellent work. I assume that the data that you presented of the higher height of children with the 2FL, that data is corrected for mid-parental height.
Is corrected for, sorry, I couldn't hear.
That the data, the results of the height is corrected, is adjusted for the mid-parental height.
For mom's height, you mean?
Well, yeah, because, I mean, it could be that the same way that genetics plays a role in determining a higher 2FL might determine that the parents are taller. And that's the child is tall not because of the 2FL, but because of the parental height.
Good point. I asked Samuli and I see him going this or that.
We corrected for maternal BMI and we corrected for birth measurements, but I don't think we corrected for maternal height, but that is an extremely important point.
Yeah, because the child's length at birth relates to maternal height, but at 4 years of age relates to mid-parental height. The father sometimes also plays a role.
I think that's a good point. Yeah.
Okay, Dr. Ogra.
These were really beautiful studies, and you presented them in such an elegant manner. Thank you very much.
Thank you.
I want to focus on the studies on the viruses, particularly rotavirus. Rotavirus, at least in the man, affects specific receptors on the terminally differentiated enterocytes. Mm-hmm. Do you know whether these oligosaccharides bind to the receptor on the enterocytes in any specific manner?
Mm-hmm. Yeah, we don't know exactly what the mechanism is behind it. It turns out, so the original hypothesis when we went in was to look for why is it that this specific rotavirus strain has a specific affinity to the neonate. Why is it that only in the first 4 to 6 weeks of life this specific rotavirus strain is found, and it doesn't infect infants or children at later age? So is there anything in the intestine? Are there glycan structures in the intestine that are different between the neonate and later on in life? So the receptor wouldn't find its binding partner anymore.
Because the disease in the rotavirus is very typically in the younger children, but not in very, very young. In the newborn period, you don't see much disease with rotavirus.
This specific virus here, the G10P11, is specific for the first 4 to 6 weeks of life.
Is that
in
right?
that specific area in India.
But you don't know whether it binds to the receptor itself.
No.
Okay. The second question is, if it does alter the interaction of the virus and the epithelium in some way, do you see any changes in the induction of immune responses
We haven't looked at that.
or expression of disease?
We only looked at infectivity in the in vitro model, and then in the cohort we looked at whether there is infection and whether there is symptoms or not. Yeah.
The last question I had was related to not a virus but a bacteria like Clostridium. It's another good example. Clostridium is an infection which you see a lot of disease in adults and old people, but not in children. In the neonatal settings, they have a lot of colonization, but you don't see much clinical disease. Do you have you looked at any other bugs besides the ones you talked about in the neonatal period?
Yeah, we've looked at a bunch of bugs. We looked, in addition to EPEC, we looked at Campylobacter, we looked at different E. coli, EACs, we looked at ETACs, we looked at Entamoeba lytica, the parasite, Giardia.
But did you look at Clostridium?
We didn't, no.
Which will be something very interesting to see, because that's another very unique setting in which this may be, you may have found a counterpart of steroids, for example. You can treat rheumatoid arthritis to viral infections and everything else. It would be fascinating. But it's an elegant study. Thank you.
Thank you. Yeah.
Dr. Hanish, and then on the third row over there.
Impressive talk. I liked it very much, and it helped me to use my impression that I'm misplaced at this meeting as a glyco person. So my question is, do we have any structural evidence on the higher mass glycans, whether they express blood group type 2 structures at all, except for the pentaoses, which are known to express type 2 structures?
Yes, so the higher molecular structures go down in concentration quite quickly.
Yeah.
We see that they may be top 15 or top 20 oligosaccharides are, at least by total concentration, the predominant players, and the moment you get larger, your concentration drops significantly. I'm not sure exactly what blood types you find on the different higher molecular oligosaccharides. Depends certainly also on the secretor, on the Lewis blood group status, right? I mean, that really drives that tremendously. But I would imagine that recaps on the larger structures as well.
I was wondering, because we found only blood group H1 and Lewis A, B structures, so no Lewis Y, Lewis X, and all these other guys. Okay.
Last question from the third row there.
Thank you for your nice deliberation of the very critical things which would help in the baby as well as in the future predictions for the chronic diseases. So my question, have you found any difference in the malnutrited mother and the well-nutrited mother in the difference of the oligosaccharides?
Say that again. Can you repeat that? Do you find differences in?
In malnutrited mother, as well as the well-nutrited mother, have you found any difference?
Yeah, so we haven't looked at that, but there is data that came out of UC Davis together with Jeff Gordon's group as well, where they specifically looked at moms. I believe it was in Bangladesh, but I think they also looked with Andrew Prentice in the Gambia, and there were differences in oligosaccharides, in oligosaccharide composition in moms that were malnourished. Yeah.
With that, I would like to thank Lars very much. Thank you.